Okay, a little refresher from where we left off. We left off talking about how intracellular pathogen gets degraded, gets loaded onto an MHC class 1 molecule, moved to the surface. That peptide is recognized by a CD8 positive T cell. CD8 is going to recognize MHC class 1. The T cell receptor alpha beta chains are going to recognize the tiny little peptide fragment. With extracellular pathogen, it's brought in through phagocytosis, and it will then be processed, loaded onto an MHC class 2 molecule, moved to the surface. A CD4 positive T cell will come in. CD4 will recognize and interact with MHC class 2, and the alpha beta T cell receptors will recognize that tiny little peptide of the antigen. And so we have these complexes form, and it takes, and this is going to start initiation, right? And that's kind of just what we know. But that's not always the case. So in some cases, we could have a situation where a virally infected cell, so a cell that has been infected by a virus, has intracellular pathogen inside of it, right? But that cell dies and gets taken up by a phagocyte, which now we have three levels here. We have the virus then that initially was intracellular pathogen, but it gets taken up by phagocytosis and degraded in a phagolysosome. But because it is a viral, so it looks to that antigen presenting cell, it looks like a extracellular pathogen. But the degraded viral proteins are still going to be loaded onto an MHC class 1 molecule, even though they came through the vesicular space. This is called cross-presentation, and this is needed for viral infections because viruses are very specific in the cells that they infect. They're only going to infect certain cells, and that's a tropism. And so if they have a very narrow tropism or infect only one type of cell, it's important that the CD8 positive T cell is engaged, and the CD8 positive T cell can only recognize MHC class 1. So even though the virus was picked up through an extracellular pathway, it doesn't change that the intracellular pathogen, the virus, is loaded up onto an MHC class 1 molecule. So cross-presentation is another way that cells, antigen-presenting cells, can be used to engage CD8 positive cells. Okay, I probably should have included that in the last lecture. It just was getting so long. Okay, we're going to move into our last concept here, was MHC expression. Major histocompatibility complex. These are human proteins that are coded for in normal genetics fashion. So you have a set of genes for your MHC based on your parents and what they gave to you. And they essentially, MHC class 1s are going to be expressed on virtually any cell, because any cell can be infected by a virus. And so then this enables the destruction of any type of cell infected by a virus to be killed, by a CD8 positive T cell, or by an effector T cell, or a cytotoxic lymphocyte, or whatever name you want to call. So our class 1 molecules are found, yes, on T cells and B cells and macrophages and dendritic cells and neutrophils. They're found on epithelial cells, because all of these can be infected by viruses. MHC class 2 molecules, though, are only found on antigen-presenting cells. And these cells are very specialized in what they do, and that the fact that they interact with CD4 positive T cells. Only a certain subset of cells do that, and that's these guys here, are B cells, macrophages, and dendritic cells. Now, they're going to have dendritic cells and macrophages are going to be a little different than B cells in the way that they interact, but nonetheless, it's still all through MHC class 2 and CD4 interactions. And so we call these 3 cells not just antigen presenting cells, but rather we call them professional antigen presenting cells. Just, adds another level of importance, right? Okay, I want to just go a little bit more into depth with the genetics behind MHC. So the major histocompatibility complex is not only involved in, well, it's in the immune system, right? But it's just a set of proteins that make you, you. So these are considered self-proteins as well, right? And they're all in this closely linked cluster of genes. So that's why it's called the complex, major histocompatibility complex. Now, histo means tissue, compatibility. Okay, if a tissue is compatible with another tissue. The thing is that these sets of proteins were looked at and explored in 2 different ways. One way was on all the tissues of the body. Okay, so these MHC molecules were found, and they're like, okay, well, these are the tissue cells. This is what makes a tissue self. This is what makes a donated tissue non-self, because these proteins are different. But these were also looked at on the white blood cells, and because they were proteins that were associated with white blood cells, they were called the human leukocyte antigens, HLAs. Well, it didn't take too long for people to recognize that the HLA complex and the MHC complex were actually the same thing. And so human MHC molecules are also called the human leukocyte antigens, because antibodies used to identify these molecules, these MHC molecules, then reacted with white blood cells, but not with red blood cells. That's where the HLA, the leukocyte antigens, came in. So just know that when we talk about MHC molecules in immunology, we're also talking about HLA molecules in histotyping. I know. I know. When we do tissue typing for transplant, we usually refer to the term HLA when we're talking about the immune system response and getting antigen recognition. We typically use MHC, but just know that MHC and HLA are the same thing. And the diversity that an individual has with MHC is going to be much less than what we see with antibody or T cell receptor, but the specificity isn't as tight. And so that's great. That helps a lot. So the HLA complex really contains about 200 genes. There's going to be a variety of different functions that are involved in there. Because there are so many, that does allow for a higher amount of specificities for binding peptides. But because of the promiscuity, that means that even though there's probably, a few hundred different specificities, it can bind millions and billions of different types of antigens or different segments of antigen. So it's really great for the human population to have this diversity, because that means that the immune system is strengthened across a population. The downside is that because everybody's HLAs are different, it makes it hard to find compatible tissues for transplant. And so a major danger with solid organ transplant or some tissue transplant is rejection. Repertoire might look too different, and be attacked by, 'cause that's what the body is recognized as non-self. So when donors are selected, it's through HLA typing or MHC typing. So it's great to have the diversity, except for when you need to do transplants. So anyways, that is just a little bit on MHC, a little bit on HLA, amount on T cell specificity. So let me know what questions you have. Otherwise, we will just keep marching on.